So I didn't bring a lot of introduction on what is the splicing process. The only thing that you should all bear in mind, and that was very clear in John's talk yesterday and this morning, is that apparently the splicing is a very simple process. So you have an RNA that is transcribed, the pre-mRNA that is transcribed by polymerase II. It is divided between the exons and the introns. To get the protein translated, all you need to do is really to get rid of the introns and join all the exons together. And then the mRNA is exported and translated in the cytoplasm. In practice, what you now know is that recognizing the introns and the exons is a very complex process. And not only the basic consensus splice sites are important, but also there are a whole series of factors which act sequentially and combinatorially to decide whether an exon will be included or not in the final transcript. And these factors are represented by auxiliary splicing regulatory elements that can be found both in the exon and in the nearby intronic sequences, but then you have other factors that can act at the level of RNA secondary structure, and RNA secondary structure, as we will see later on, can either promote or hinder the recognition of the splice sites. And then, of course, there are a whole series of factors that are very hard to predict beforehand and have to do with the velocity of the RNA pol II, so the velocity at which the pre-mRNA is transcribed, if there are particular splicing events occurring before the exon that actually has to be included or not, and also external stimuli or just the general status of the cell. So basically, because the process is so complex, it is really not very surprising that a lot of splicing mutations end up in connecting with human disease. And splicing mutations, I'm going to talk mainly in this first part of the talk about splicing mutations that occur either in the consensus splice site sequences or in the nearby exonic and intronic auxiliary elements. And if you look at splicing mutations in all the genes that are subject to alternative splicing and which are connected with disease, as John pointed out, you have a quite impressive number, because depending on the genes, of course, some genes are more susceptible to splicing mutation than others. Depending on the genes, the frequency of splicing mutations that can account for the disease range from as low as 3 or 4% up to 33 or even 50% for genes like ATM or CFTR. And if you think about the problem of screening with splicing mutations, really a lot of medical people, what they are concerned is, to get a indication by researchers like us, whether a change that they see in these consensus splice sites on these auxiliary elements is really a splicing mutation or not. And what these splicing mutations do, basically, in our experience, you have 4 major consequences. So if any of those following a mutation, the most likely event that can happen and that we see happening in patients is the skipping of the exon. So the exon is completely ignored by the splicing machinery, and this often ends up in a change in the reading frame and with consequence degradation of the resulting mRNA by NMD. Then sometimes the second most likely outcome of a splicing mutation is represented by the activation of cryptic splice sites. So let's say that our splicing mutation inactivates the 5' splice site, then sometimes the splicing machinery is still able to recognize the 3' splice site that is kept in its natural status, and instead of using the original one, which is now mutated and doesn't exist anymore, it will simply pick up alternative 5' splice site sequences that are normally ignored, but which in this case are activated. And also in this case, the most likely outcome is that you have a change in the reading frame, or if you are extremely lucky, you have just the addition or the deletion of amino acid, which could still end up in disease if they occur in a very important part of the protein. So these are the two major consequences of splicing mutations that we see associated with disease. Other consequences that are quite present are intron retention. So if either the 3' or 5' splice site become inactivated, then the splicing machinery simply forgets to process either the upstream or the downstream intron, and these introns are included in the final mRNA, of course, most likely introducing stop codons and that kind of stuff, which inactivate the protein function. And then another quite common, because people have now started looking for it specifically, another quite common consequence is that when mutations fall outside a known exon or in the nearby intronic sequence, they sometimes can actually create new 5' or 3' splice sites. And when they create new 5' or 3' splice sites, very often the splicing machinery actually is able to pick some cryptic splice sites around this de novo splice site, and ends up including a sequence, an intronic part of the intron that is now called a pseudoexon. So this pseudoexon, and these pseudoexons, of course, very often end up in introducing again stop codons, changing the reading frame, and so on, and so they disrupt the functionality of the protein. So how do we, in the medical field, how do we pick up these mutations? And there are basically two ways in which this is done. Here I'm showing three because but in vitro splicing now is getting a little bit outdated, especially because you have to use radioactivity, and not many clinical laboratories like to do that. So the two ways that people use to screen for splicing mutations associated with disease are, of course, the obvious one. You go and look in the patient, this has the obvious advantage that if you look at the patient, you see exactly what is going on. There are some disadvantages, and basically because sometimes the patient is not available. If you screen for splicing mutations for some genetic diseases, it's not like you have many people who are affected by the disease. Maybe they are spread over the entire Europe or in different countries, and so it is very difficult to actually go to the patients and see what is the splicing, what is the defect in the splicing process that they may have. So then, of course, the other issue that is quite important and that very often prevents from looking at directly at patients is that maybe the gene where the splicing is affected is not expressed in the available tissues. Just to say, what one usually what is easy to obtain from patients are blood monolymphocytes. So if the splicing defect is occurring in a gene that is not expressed in blood cells, then of course it is very difficult to get the material on which you can do the RT-PCR analysis and see if there is a defect there. And then of course what happens is that when you do this kind of approach, you always have to take care of the fact that many of these splicing mutations are present only on one allele, and so you have to take into account when you do an RT-PCR approach to see the defect that there is also the other allele being expressed as well. So in order to kind of avoid all this, what we normally do, and what is done now in many labs around the world, is a kind of a mini-gene approach. So you actually make an artificial pre-mRNA where you insert the exon where the clinician thinks that there is the splicing mutation is occurring. This mini-gene is transfected into cells, and you can just see using RT-PCR whether the exon either following the mutation or if there is a difference between the inclusion of the exon in the wild type when you have the wild type sequence or when you insert the putative splicing mutation. And I'll just give you some examples of how this can be applied to a few diseases with clinicians that we are collaborating with. So this work is done in collaboration with Diana Baralli in Southampton, and she has been studying Marfan syndrome. Marfan syndrome is a relatively common autosomal dominant disorder that that can occur when you have mutations in the fibrillin gene, and what happens is that of all mutations described in the fibrillin gene described to cause Marfan syndrome, 13% actually have to do with splicing. So what we did together with Diana was to really set up a series of primers that amplify selected region of the gene. And the gene is expressed in monocytes. So if you want to screen for splicing mutation in Marfan, in potential people affected by Marfan syndrome, what you do is you simply extract the RNA from their lymphocytes, and you actually do an RT-PCR using all this set of primers, and you see if there are differences in the amplified fragment with respect to what you expect from a normal control. And indeed, like you see here, using this approach, for example, we have been able to identify some variations. We were actually screening for variations which could be putative splicing mutations or could also be just harmless polymorphism, and out of all putative splicing mutations, we actually identified a new one that had never been described before. Now, this of course cannot be applied, as I said, to all the diseases that you might be interested because the patients are not available or because the gene is not expressed in the blood cells. So what you have to do is you have to actually use a mini-gene approach. And in this mini-gene approach, just to show you an example, we have used it to actually screen for neurofibromatosis mutations. So neurofibromatosis is another inherited disease that gives a lot of problems at the musculoskeletal, ophthalmologic and cardiovascular level. It is characterized by the appearance of these café au lait spots, which are tumors, small tumors that actually appear all over the body. And so, for example, in this gene, we have concentrated on mutations that affected the recognition of a particular exon that is particularly affected by this kind of mutation, and is exon 37. So you see, for example, mutations in this exon have been long known to be associated with disease, and you see, for example, in this normally in this exon, as part of this exon, you have this sequence here where TAC codes for a tyrosine, and in the past, the previous people who were screening for patients actually found that this C was changed into a G, so tyrosine was changed into a stop codon, and so this mutation was kind of explained that the functional consequence of the mutation was explained as this change was C to a G was actually changing from a tyrosine to a stop codon. But when you put the exon 37 sequence inside your MiniGene and you put it either in the wild type or in the mutant configuration, you see that not only a stop codon is being created at the protein coding level, but also the amount of exon 37 that is normally included in the MiniGene, when you change from a C to a G, you actually have a complete skipping of the inclusion. And this is quite important if you want to look at this issue from a diagnostic screening point of view, because for example, you see, this is a completely artificial mutation, so this has not been described yet, so then instead of having a C, we replaced this sequence with a T, so the coding capacity of this triplet was unchanged because they both code for tyrosine, but even when you change, when you do this change, you also have skipping of the exon. So this is quite important from screening and diagnostic purposes, because if you think that this change TAC or TAG only had an effect on the protein coding potential of the gene, then you would, when you encounter a patient that has TAT, you say, well, this is a harmless mutation because TAC and TAT code for the same amino acid, but actually this patient could actually, the phenotype of the patient could actually be explained by changes in alternative splicing, because TAT is actually causing the skipping of the exon. So having said this, you can also use the MiniGene system to look, for example, in this case, for genes that are not normally expressed in the blood. In this case, we were asked for help by people who were working on the long QT syndrome. So this is a syndrome that often occurs in patients that have a heart condition that is associated with the prolongation of the Q and T waves in during their heartbeat. And what happens is that these patients have a prolongation of these waves, and since this is a situation that tends to worsen over time, they often die from a sudden death because they have a heart attack. And of course, it is very difficult. You know, if you look at patients that have this condition, it's not like you can take very easily a biopsy from the heart and see if there is a defect in splicing. And in this case, the putative splicing mutation was also very strange, because it was not really occurring in the basic consensus elements, so that people immediately would have been able to say, well, yes, this is probably a splice site mutation. It was occurring at position +6, which is of the 5' splice site in intron 7. And this position can vary quite a lot, because you see that if you look at the frequency of the nucleotides in the 5' splice site, you see that whilst the position +1 and +2 are almost always GU, and this is conserved almost 100% of the times, if you go down to position +6 of a donor site, then in 41% of normal individuals you can find a T, and in 21% also of normal individuals you can find a C. So it wasn't very clear if a patient that carried a change from T to C, this was really was a change capable of affecting splicing, or it was just a harmless polymorphism. And you can go and see the heart, what happens in the heart, but what you can do is you can do a MiniGene system. So you just get your exon 7 with the introns, part of the introns, both upstream and downstream, and you make two MiniGenes. One MiniGene that carries the wild type sequence, so a T in position +6, and another one that carries a the C, the putative splicing mutation in position +6. You transfect the mini-gene, you do an RT-PCR analysis, and you look what happens. And you see that if you have the wild-type sequence, what you do is you get complete inclusion of the exon, but if you introduce a C, then you have intron retention. So this entire intron is not processed anymore and is kept inside the mature mRNA. And this actually, so you can prove that indeed this change here is affecting the 5' splice site because, as John told you yesterday, the 5' splice site is recognized by U1's NERP through a base pair interaction, through a RNA-RNA interactions, and normally this T in position +6 base pairs with the A present in the U1 small nuclear RNA. When you have a replacement of a T for a C, this base pairing cannot occur anymore, and so one would suggest that in the presence of this mutation, U1's NERP would not be able to recognize the 5' splice site any longer, and this was proven by us because what we did was to make a mutant U1's NERP that instead of carrying an A in this position here, it carried the G. And the G in position +6 in U1 small nuclear RNA now was predicted to be able to base pair with the mutant C in position +6 at the mRNA. And when you transfect this mutant U1's NERP together with the mini-gene, you see that you recover correct processing of the exon 7, and you have a big decrease in the amount of intron retention that you observe in your assay. So, so this is just two examples to tell you how mini-gene can be very useful in spotting and in actually validating putative splicing mutations. Mini-genes can also be used to check for effects that not only affect the basic splicing elements, but also mutations that affect the RNA secondary structure. Just a brief introduction now, because very often in the literature you will always see a pre-mRNA shown in this way, so like a kind of linear sequence where you have your exons, the cap at one end, the poly A at the other end, and but this is really something that will very seldom happen in the cell, because if there is one property of RNA, and Dino was talking about this during the first day, if there is one property of RNA that distinguishes it from DNA, is the fact that RNA really likes to fold upon itself and to create a lot of structures, secondary, tertiary, and quaternary structures, and these structures can have quite a huge effect on splicing. Normally, these structures are kind of kept in check by the presence of proteins that are binding to the pre-mRNA, and these proteins start to bind as soon as the pre-mRNA is transcribed. And these proteins normally belong to the H9P family of proteins, and what these proteins do is kind of keep in check this tendency. And so what you can think whenever you think of mRNA, what you think is a situation where the mRNA would normally tend to fold upon itself in a very tight manner, but because these proteins are binding to it, then the number of structures that the messenger RNA can form is also a little bit limited. So you are in a kind of hybrid situation. But this hybrid situation is very useful because it can be used by the splicing process to actually control how splicing is the efficiency of splicing. So and it can do this in quite a few very simple and intuitive ways. So for example, you see here, if you have a 5' splice site that is embedded inside a stem element, then of course you can well, imagine that this 5' splice site will not be very able to interact with U1's NERV, and indeed, if you do this, this is a completely artificial system where you increase the strength of the stem element, you see that the use of the 5' splice site is actually inhibited the stronger the stem element. But RNA secondary structure, of course, is not just inhibitory, it can also help the recognition. So, for example, if you have different branch point sequences that are maybe too far away from the 3' splice site to have an efficient recognition, if you have the formation of a stem element in this between the branch point sequence and the 3' splice site, then you can actually promote its use. And also you can do this by getting nearer of or enhancer element, various enhancer elements that really have the same purpose. These are just other elements of this. And just to give you a connection between, this is not a connect, because it's not very easy to look at RNA secondary structure. This is not something that people normally look at when they look for a connection between splicing mutations and disease. There is a lot of progress that is being made. But at present, I think the best example of this is given by the tau gene. So in the tau gene is quite important because it's connected with Alzheimer. You know that mutations in the tau genes are often associated with frontotemporal dementia and Parkinsonism. And very often what is important for the tau gene is the recognition of this tau exon 10 in a limited quantity. So the tau exon 10 must not be very highly recognized, because this brings an imbalance in the isoforms of tau exon 10, and this imbalance can eventually lead to disease. So in order to have I would say, limited recognition of tau exon 10, what nature has done is to actually embed the 5' splice site of tau exon 10 into a stem loop structure. And this stem loop structure is actually competing for, so this is the 5' splice site, this is the stem loop, and the stem loop structure is competing with the binding of UVA small nuclear RNA, and this stem loop structure is quite strong enough to actually give the correct ratio of exon 10 inclusion and skipping in the final mRNA. Now, in many people who are affected by frontotemporal dementia and Parkinsonism, they have mutations that disrupt the RNA secondary structure, and so they make the 5' splice site more available for the splicing process. And when you have this situation, then the final mRNA will end up with a much increased ratio of exon 10 inclusion, and this will eventually lead to disease. So just to finish up a little bit on these, these are the direct connections that normally people who do research in alternative splicing have with clinicians and medical people who are not very much interested in the theory, but they are very much concerned with saying this is a disease causing mutations or it is just a harmless polymorphism. And so these are the practical approaches. Of course, as you can well imagine, because our ability to sequence patients has grown enormously during recent years, because the cost of sequencing a gene has dropped by a huge amount in recent years. What happens is that now we are actually invaded by a huge number of changes that are provided by clinicians, and they are interested in evaluating all these changes and deciding whether these are mutations that could affect splicing or not. And of course, it is very difficult to do this either using a MiniGene or just looking directly at the patients. And so a huge amount of effort in recent years has been dedicated at bioinformatics approaches that could be useful in predicting splicing mutations. And there are now quite a number of freely available applications that you can simply connect to the internet, and these applications are all aimed at predicting splicing mutations. Some of them actually will predict splicing mutations at the level of the basic splicing elements, like the 5' splice site, the 3' splice site, or the branch point. Others also look at the possible disruption of these enhancer and silencer sequences that Silvano talked about and John too. And then there are also a few that now have started looking at RNA secondary structure. Now, I cannot really possibly go through all of them, but of course, if you are interested in this kind of work, you can get the list from my presentation that will be provided to you in the CD. Just let me show you one of the latest one and what you can expect. So this is Rugol. It has been developed by Gil Ast in Tel Aviv, and what it does, it kind of combines a lot of these bioinformatics approaches together to make the analysis a little bit simpler. What you do is, if you're screening for splicing mutations in a particular gene, you just input the sequence of the wild-type sequence, you input the sequence that you think carries a putative splicing mutation, and the program will actually give you a readout where both for the wild-type sequence and the mutated sequence, it will give you an idea of how strong is the 5' site, how strong is the polypyrimidine tract, the 3' and if there are enhancer or silencer elements, and you can see, without actually doing or performing any work, you can actually see if your mutation is predicted to affect some of these. Your mutation then scores positive in this kind of bioinformatics approaches, then you can actually think about getting the RNA from the patient if possible, or if not, you can decide to go for a MiniGene analysis. Now, how good are these programs? Because of course this is I think the most important questions. They are, I would say, fair in a way that they still miss a lot of enhancer and silencer sequences because they are very difficult to predict. Just to show you an example from our lab, we do a lot of validation of this kind in our laboratory. So if you look at the fibronectin ADA sequence, we know that this of the exon, and there are no other regulatory elements in anywhere else. So if you take the sequence of the entire exon and you put it through these programs, you see that really the programs predict the presence of enhancer or silencer sequences a little bit everywhere, with perhaps just a little bit of a prevalence on the real enhancer. So there is still a long way to go, but at least if you have a huge number of mutations and you want to prioritize which mutations you are going to look at in a MiniGene setting or directly in the patients, then these mutations these approaches are, you know, quite useful. The last two things I want to say about these basic connections has to do with M-fold and P-fold, the RNA secondary structure and prediction programs. Everything has been actually being talked about by Harry in a beautiful way just yesterday. There are basically two ways that, if you think that your splicing mutation is affecting RNA secondary structure, there are two ways that you can go about testing it in a bioinformatic way. You can use energy minimization programs, the most famous of which is M-fold, and that is the one mostly used, or you can go through P-fold, that is a RNA secondary structure and prediction program that uses a sequence comparison. So you need to know the homologous sequence in many organisms, and then the program will align it together and will give you just one consensus output. As Harry very correctly said yesterday, there are advantages and disadvantages of either approaches, but in the end, I must say that they are not. It is still very difficult to predict whether your mutation or not is actually affecting the RNA secondary structure. So what is the conclusion of this first part of the talk? The first part of the talk really is that now people have started to draw up some protocols. If you look for splicing mutations connected with disease, then of course they give, this is the kind of advice that you are usually given and that is summarized here. So this may sound a little bit trivial, but check the literature. Now it's very easy to do. Very often maybe your variant has already been experimentally tested, and so that will save up a lot of your work. Perform the bioinformatic analysis. Again, the value of bioinformatic analysis can vary can be quite variable, but still will give you an information on where to prioritize your efforts. And then, of course, you need to go through the experimental validation, and then eventually you can actually reach a conclusion. So just a small few in the last final 5 minutes of this first part. So just a few words about what do we do with all this information, right? Once we know that splicing mutations are important for disease, now we know how to detect them in a huge number of patients. Things are getting better, so what do we do? And really, this is something that I think will very much change our ability, because the research in splicing will very much improve our ability to treat patients affected by a number of diseases, because as Silvano mentioned towards the end of this talk, there is now a real possibility of actually affecting the splicing outcome of particular exons, and of course this possibility can be used to improve or to cure kind of diseases. This is just some of what I think are the most significant examples from a beautiful review by Garcia Blanco a few years ago. Now there are several other approaches, but I think most of them are, but the approaches that I'm telling you are the ones that are actually being tested for efficiency in a clinical setting. So for example, if you have people that have splicing mutations which activate cryptic exons inclusion in the final mRNA, then what you can do, you can use antisense oligos to actually block this inclusion, and you restore normal splicing. What you can do also is, if these inclusion create a particular or they add new exons or part of an exons that you don't want to your final mRNA, what you can use, you can exploit the fact that you can silence these particular inclusion events using specific siRNAs that target the splicing event that you want to get rid of. What you can do is, if you have, instead of acting on the mRNA, if you have, in this case, this is the SMN2 gene, if you know that your mutation is affecting an enhancer element that decreases the efficiency of binding for some particular splicing factors, then you can use compounds that increase the amount of splicing factor made by your cell. And because, as you can as I hope you all know, the binding of a protein to an RNA is also subject to a chemical equilibrium. If you increase the amount of transacting factor being produced by the cell, even it is possible that this transacting factor will then be able to bind more efficiently to the mutated sequence and therefore restore correct splicing functionality. Then these I won't talk about because they are these TOS and TOS element Silvano just said. And the last thing I want to finish this is that splicing therapy, of course, can also be applied, for example, to changes in RNA secondary structure. In this case, you see the example I've shown you before, so the tau exon 10, this is the tau exon 10 stem loop, and mutations that weaken this stem loop have the ability of causing an increase in the 5' splice site usage. So if you can model this stem loop through X-ray crystallography or NMR, then you can actually start to think about making molecules which can stabilize this stem loop also in the presence of mutations, and therefore you can recover correct 5' splice site usage in people who are affected by disease. So this is just the conclusions. What we need and what we start to have is a better knowledge of splicing. That is always important, because if you know what is affected, then you can take advantage of this for therapy. Of course, the better bioinformatics tools, this is something that many people are working on, and they are growing steadily better. And then, of course, better splicing tools, which will make, for screening and diagnostic purposes, our life much easier. So I think I'll stop here for a few minutes so that you can ask questions on this part of the talk, and then we go on with the second one.